It's my pleasure to introduce Susan Dutcher from WashU, Professor of Genetics at WashU, to talk about cilia. And I fondly thought, until I talked to her at the coffee session, that there was a relationship, A, between cilia and flagella, and I was told that there isn't. And secondly, I thought that otoferlin, the gene therapy that is being used to treat deafness, was treating a ciliary defect. Well, it kind of is in a way, but it's a different kind of cilia than the cilia we're going to hear about. So without further ado, let's hear about it.
So I would like to thank the Larrys for the invitation, and hopefully I can show you some of the enthusiasm about cilia. There are these little, whoops. There are these little organelles right here coming off of this organism, which is called Chlamydomonas. They're present in all the organisms that are here on the slide. And I wanted to start by telling you what the title here meant, from pond scum to human disease. So when I first got to Washington University, I was very excited. The dean of the medical school asked to talk to me, and I thought, oh, this will be really fun. And so the dean called me in and he goes, "Why did we hire you? You work on pond scum. Why are you here at a medical school? This has nothing to do with human disease." So it's true, pond scum does not cause human disease, but what we've been able to do over the last 25 years is to show the importance of these organelles, of cilia, with respect to human disease. And the thing that makes it possible is because cilia are incredibly conserved, they're evolutionarily conserved, and we can go from many model organisms to use them to actually find genes that are actually involved in human disease. And those diseases are wide-ranging. They are diseases that affect the eye, the kidney, the liver. They cause short bones. I'm going to tell you mostly today about their effect in the respiratory tract. They cause laterality defects. So instead of having your heart on the left side, your heart ends up on the right side. They cause all kinds of heart disease problems in which children are born with malformed hearts. They have major effects in the brain in terms of degeneration and other kinds of things. And they actually are involved in obesity, mostly by affecting your ability to control your appetite. I can't have done all of this without an amazing group of people. I have great colleagues that actually work on various aspects of cilia, and we have colleagues that range across the medical school, colleagues that do neurosurgery to colleagues that do biochemistry. And in particular, Steve Brody, Amjad Hirani, Phil Bailey in mechanical engineering, and Ray Zhang in biochemistry have really made some of the things that we do possible. And as Sean said, we couldn't do what we do without the money that comes from NIH and from NSF. And so I feel really lucky as a person who works on pond scum, which is supported by NIGMS, but also to have money from heart, lung, and blood to actually take the discoveries that we make in pond scum and algae and actually apply them to patients. And I can't do this also without the people in my lab. So Chlamydomonas is the organism that we work on. Come on. Sorry, the movie didn't go. Anyway, this is a cross-section by electron microscopy looking through a cilium. So if we cut across the cilium, it has all of these structures in it. Unlike the bacterial flagella that you heard from Sean, which has in the flagellum itself one protein, the eukaryotic cilia probably have somewhere from 700 to 900 proteins that make up this incredible molecular machine. And so if you think about it, the proteins that are actually in the cytoplasm, as well as the proteins that are actually out here in the cilia, make up probably about 7 to 8% of all your genes in your body, in your DNA. And so they are a major organelle, and yet for a long time they were sort of underappreciated. And as the movie, see if I can go back, because I'm having, there. So right here I wanted to illustrate to you a microtubule. It's a kind of a boring molecule. It's made up of alpha and beta-tubulin. It doesn't have a lot that would make you excited, but they're the key to building cilia. They're the things that make up all of the structures here that are the 9 doublet microtubules with all of these additional proteins. And the motility that cilia have is organized by this motor, which is called a dynein. A dynein is a huge molecule. It's about 2 million Daltons, and it walks along microtubules. And that walking then pulls other things along and then gives us the motility that you actually see. And I should say that the little organism we work on was found in a potato field in Amherst, Massachusetts. So I'm now showing you the part of the tree that Sean showed you was a very small part of life on the planet. This is the eukaryotic branch of the tree. And the last common ancestor down here, we think actually had motile cilia. And we took advantage of the fact that across this whole spectrum, there are 2 examples. One in the flowering plants and one in the fungi, in which cilia were lost. And so all of the other organisms in the eukaryotic tree have motile cilia, with those exceptions. And so when the genome of Chlamydomonas became available, Jen Billy Lee, a graduate student in the lab with help from Gary Stormo, did a multiple sequence of an alignment between Chlamydomonas genes and human genes. And we found that there were about 4, I'm not having good luck with this. There are about 4,000 genes that are conserved between Chlamydomonas, the green alga I showed you, in humans. But what we were really interested in was to take advantage of these 2 organisms having cilia and subtract the genes that using Arabidopsis. And so we wanted to find things that were in Chlamydomonas and in humans, organisms with cilia, but missing in Arabidopsis. And when we do that, there's about 700 things come out. So this is a paper from about 20 years ago. We now know that those 700 genes that we predicted by doing alignments between the 2 organisms and then subtracting genes that were shared, all those housekeeping things that are in Arabidopsis, to give us genes that were actually in cilia. When we started to look through that list of genes, one of the classes of genes that came up very frequently were a set of genes called Bardet-Biedl syndrome, BBS genes. And these are patients that have central obesity, they get retinal degeneration, they have kidney disease, and they have mental retardation. And the papers about these things all said, these are genes involved in energy metabolism. They probably affect mitochondria. And when we looked, and they were in Chlammy, in our set of things that we thought were massively enriched in cilia genes, we said, no, BBS is going to be a disease of cilia. And so we looked, we contacted a group that had actually had a family in Newfoundland that had Bardet-Biedl syndrome. They had mapped it by the old techniques to a region that had about 250 genes. We compared our set to the 250, and we came up with a gene that we now call BBS5. It was in this family. It's an autosomal recessive. And then we looked at other families that had Bardet-Biedl syndrome, and we found 3 other mutations. And so we now know that the BBS genes are involved in a complex that's transported up and actually determines the membrane protein composition of cilia. And things like neuropeptide Y and leptin receptor are receptors that actually live in the cilia in the hypothalamus. And so we went from Chlamydomonas and looking for genes to actually finding genes that were involved in a variety of human symptoms. We then were really amazingly helped by a conversation over coffee with a junior colleague. Ray Zhang was a relatively new assistant professor in the biochemistry department, and he had come to show me his pictures of proteins that bound to microtubules. And I said, well, how about we use your technology to look at cilia? And his technology was to use single particle cryo-EM. So single particle cryo-EM basically uses samples that are rapidly frozen. You then take pictures and average over them, And people had done this to get amazing kind of structures of all kinds of things. And generally there are relatively small things. There are single proteins or complexes of proteins. But I wanted to do it on cilia because we lived in the world of blobs. So this is a picture of cilia from Chlamydomonas. Here's a wild type. Here's a mutant. And you can see here, looking longitudinally, that these blobs are missing in this mutant. And I thought, let's go from being able to see blobs to maybe being able to see a whole lot more and understand more about what's going on. And Ray said, well, we can't do that because to make it work, the things have to be small. They have to be 25 nanometers. 25 nanometers, for those of you who don't think in that world, is 1/1,000,000th of an inch. So we had to get something that would let us do that. And this is 250 nanometers. It was 10 times too big. And I said, I can solve the problem. We can do it. And the way we did it is we took Chlamydomonas, we treated them with a pH shock, makes the cilia fall off. We then treat the cilia with a little bit of ATP and a little bit of protease that will cleave a connection between each of these 9 microtubules. This is it right here. So now each of these lines of dots that you see is a single doublet microtubule. Now they're only 25 nanometers thick. We froze them. Here they are. Here's a doublet microtubule frozen and averaged 100,000 images. When we do that, you start to be able to actually see proteins. We can see structures. And these are the 39 proteins that we were able to see within the doublet microtubule. There are long sorry, I have a real problem here. There are long filamentous proteins. There are ones that are quite globular, and then ones that are slurred or disorganized. And the thing that was actually amazing was that you can begin to actually read off the amino acids. So not only did we actually find out what proteins were present, we could actually see what the amino acids were and identify each of those kind of things. It means then that you actually can be able to tell what proteins on here bind to the microtubule, what proteins in this purple one bind to the orange ones. You actually get protein-protein contacts for this machine now that we can actually begin to think about dissecting out. So here are our proteins. We love this set. And then we wanted to know what they did. So let me just show you. I showed you here in cross section the proteins, but we can also look at them longitudinally. You can see these long ones that are running. They're actually 48 nanometers long, and they actually bind to various other proteins. But it meant we actually had amazing resolution. Something I thought we would never be able to see is that you could actually see these incredible patterns. We also then wanted to see what happened. We call these microtubule inner proteins. And so we actually made mutants. These are the Chlamydomonas plates, and we picked mutants from them. Here, let me just show you 2 of these. One is FAP166. It's this green protein right here, right here. When you make mutants, that green protein goes away, and that's all that happens. In the RIB72 mutant, which is this orange one right here, it has a much bigger effect. And so now, instead of losing just RIB72, we lose 3 more proteins. And so we can actually map out these phenotypes using the single particle cryo-EM. We wanted to then know, what do they do? And so I'm going to tell you about these guys. Here's each of the proteins. They're all living inside the microtubule. And we spent a long time looking at how they swim under low light. And so Chlamydomonas can swim forward. It uses this, what we call breaststroke or asymmetric swimming, and it pulls the cell along. And it does that to swim toward light because it's green and it does photosynthesis. But if it's in really bright light, They don't want that because it bleaches all of their chlorophyll and their membranes, and so they actually change their waveform, and they make a symmetrical waveform, and they swim backwards. And so we mostly were thinking about forward swimming and breaststroking along, and we looked at all of these mutants. They were completely wild type. We looked at every parameter in the world. They didn't do anything. But one day, a postdoc in the lab, Mathieu Bautier, says, why don't you look at reverse swimming? So we hit them with bright light, and wild type swims backwards. These guys are all immotile. They just stop. And the mutant I showed you, FAP166, doesn't quite stop. It's swimming backwards, but it is about 1/10 the rate of normal. Now, that was really interesting, but then we realized that the genes that are the orthologs of the clammy mutants are shown down here below. And these are genes, when the variants are examined in human patients, have two phenotypes. They affect the sperm. These men have immotile sperm because the sperm looks like it requires these proteins. And they have laterality defects. So during development, there's a region called the node that has motile cilia that swim like this. And it actually makes the decision of the embryo developing which side will be right and which side will be left. And so these patients then have reversed laterality. And so we were really excited that, in fact, we had a fairly good idea that these MIPS were not affecting swimming forward, they weren't affecting length, they actually were affecting the events that happened both in the sperm and in the node to make the ultimate decisions about left-right asymmetries. I liked this paper that came out in 1986 that said maybe we could think about using humans and not just model organisms for thinking about what's going on, because in the human population we have a wide range of variants that actually have been saved. And so it was at about this time that we started thinking about not just looking at Chlamydomonas, but trying to think about a little bit more about what was going on in humans. Let me tell you that there are two kinds of cilia. There are sensory cilia that are on almost every cell in your body. They serve as little antennae. They receive signals via receptors, often through G-protein coupled receptors. They also receive ion signals through ion channels and calcium receptors. And they also send signals by making ectosomes. I'm not going to tell you much more about sensory cilia today, but instead I will tell you about things that are actually going on in the ependymal cilia. Those are the cilia that are in the third and fourth ventricle of your brain. They're sitting there mixing up the cerebral spinal fluid, and the cilia that are in your respiratory tract. And at that point, we started to think about a disease that's called primary cilia dyskinesia. That means that the primary defect is the cilia have altered motility. And we ended up with this family that came to Amjad Hirani's clinic. He was a young physician scientist who had just arrived at WashU. And he had patients that had come from this town called Buffalo, Missouri. It's a very large family. And they had 9 kids when we first started. They now have 29 affected kids in their family since we first looked at this. And we sequenced the 9 affected kids. These are kids who have newborn respiratory distress. They almost always need oxygen when they're born. They actually can have laterality defects. They always have their noses obstructed. They often have cases of pneumonia. As they get older, they're always coughing. They're always coughing. They always have a runny nose. Some of them actually lose hearing because of infection in their ears. They have bronchiectasis, so they lose lung function. And those all continued, and then they're actually also associated with male infertility as the men get older. We sequenced these genes, and we ended up finding a gene called HEDAR2. And at the same time, we're looking at a family from Israel, and it turned out to be a gene called LRC6. These two genes are together do an interesting thing. They have to help build dining arms. So I showed you the picture of dining walking. I told you dining is really big. And it turns out that it is actually made in the cytoplasm as the whole 2.5 megadalton complex. And it's made in little factories, which have been called dynaps, and they're shown here in little yellow dots, in which all of the proteins that go into these actually assemble and fold in here, and then are transported by this machinery from the cytoplasm out into the cilia, where they actually find their home and act as dinings to move the motors. And we like to think of it a little bit as a factory, that we have these factories that are assembling these structures in this cytoplasm. At the same time, the Chlamydomonas, the green algal part of my lab, were working on mutants that were actually isolated by Sir John Randall. He's a Sir because he actually helped in the development of radar during World War II, which is the same components that actually now are used for making microwaves. And he then decided to move into molecular biology and started isolating mutants that couldn't swim. So they were paralyzed. And we were looking at two genes called PF7 and 8, or CCDC39 and 40 in Chlamydomonas. And they had this amazing phenotype that instead of those 9 nice doublets that I showed you before, these are just messed up. And so we were really intrigued to try to figure out what was going on here. So it all sort of came together. Amjad actually had another set of patients that turned out to have variants in 39 and 40. With Ray, we were able to show that 39 and 40 make this peach or golden heterodimer that runs along the microtubule. And we were then sort of set to see if we could try to figure out what was going on. I put this graph up because the patients that have 39 and 40, they lack motility, much like the other 48 genes that have caused this disease, primary ciliary dyskinesia, but they're much worse. So they're measured by this test called FEV. So if you take a really deep breath and you expel it, all of you would probably be up in this part of the curve. This is how much air you can expel in 1 second. These patients have a horrible time. They have a very low rate of expelling their breath, and they have a hard time breathing. And the treatment that they actually undergo is that the mucus is there. They actually need to get rid of it, and so they wear these vests that thump them. This is the treatment. A little bit of antibiotic and thumping the breast to loosen up the mucus so that you can get rid of it. And so we decided to then take advantage of proteomics that was actually coming along. We had run a lot of 2D gels, and we thought that there were about 200 proteins. But with proteomics, we now know that there's a lot more. And when we looked at the proteomics from human cilia now, isolated and examined, they turned out they were missing inner dining arms. Those are the 2 red dots on the inner circumference. They're missing this structure called the NDRC that holds the doublets together. That's why the things were all messed up, because they're not held in proper register. And they were missing some of these structures called radial spokes. Now, I showed you the picture of the microtubule, and I told you it was really didn't have very many landmarks. And so we began to think that maybe this molecule 39 and 40 that makes this heterodimer might be able to begin to provide addresses. And so this is just to remind you that if you flew into Denver yesterday, you had to somehow get from Denver into the Butcher Auditorium here. And so you took advantage of addresses. And 39 and 40, we think, actually help provide addresses for about 120 different proteins within the cilia. So the first thing that happens is here's the doublets. They all look, I told you, pretty much the same. And that somehow 39 and 40 now finds the right place. It binds here on A2. It picks one of 23 places. Then there are a set of proteins called CARPs, which stand for cilia address recognition proteins. There are 11 of them. They all come and bind to particular places on 39 and 40. 39 and 40 provide a little address. These addresses are about 4 to 8 amino acids long. And each of these now bind, and we've actually done mutagenesis, much as Sean was sort of mentioning, in which we wanted to go in, we make changes in 39, the CARP doesn't bind for that site, or we make a change in the CARP, and it no longer can bind to 39. And that's because the cryo-EM tells us exactly what to mutate. We can actually now go in and hitch each of those addresses, or the proteins that bind to the address, and now show that it actually causes a loss of a structure that we thought about. And then finally, as the CARPs bind, so one is IQUB for this radial spoke, or CFAP91 for this radial spoke. Those now are the things that actually bind to the addresses. And as you can see in this image, here is the CCDC 39 and 40 in yellow and peach. Here's FAP91. And we know which ones are going to actually sit there and bind. And so this molecule now lets us begin to think about how you start to build this incredible structure and put everything in the right place. And so we did time lapse. We know 39 and 40 enter first. We know the CARPs then enter, and presumably then bind to the higher dimer. And then all those other structures come in secondly. And when we don't have 39 present and it doesn't enter, the CARPs basically enter, but they don't have a place to dock and they have to come back out, and the complexes don't come out. Now, the name of this disease is ciliary dyskinesia, and the world thinks that the defect is because they don't have motile cilia. But why, when we have 50 different genes, do 39 and 40 have such a more severe phenotype than all these other things? And I think the field was confused because we thought the lack of ciliary motility was the phenotype. What I want to tell you is that in 39 and 40, there are non-motility phenotypes, and they are what responsible for making this disease more severe in these particular patients. So I wanted to just show you this again. Did you notice they sped up? That's because cilia in these images have taste receptors on them. And so if you give them a bitter compound, it binds to the taste receptor and it changes the motility. And so I told you that there were sensory cilia and motile cilia. It turns out they actually probably do both. And so the first non-motility phenotype is that we have to clean up and recycle trash. So in 39 and 40, there's about 120 proteins that get made. They actually continually are made as we look at transcripts, and they have to somehow get removed, we think. And so we started to think about how that might happen. And so this is just to show you that this is one of the proteins that in the cilia in magenta. It's lining up with the tubulin. They're all carried up via the transport that you can see in these images. But in the CCDC 39 patient, there's still some protein down. We know that it's actually much reduced. It's probably only at about 10% of normal. But it's not in the cilia. It's down here below the cilia. And so we were trying to figure out what might be going on. We know by using turf microscopy that they actually move up on these trains, on the motion that's here, and they get returned back. So we turn to single-cell RNA-seq. So we analyze human cultures that have basal cells, which are the progenitors. They have secretory cells, and they have ciliated cells. We looked between normals and 39. They're basically the same. But there was a class that was actually massively altered in 39, and that was under the Go term proteasome. And so when everyone in this room that thinks about this thinks about it, they probably think about 26S proteasomes, which are the machinery that recognizes proteins that you want to get rid of it ubiquitinates them with this small molecule, and then ATP comes along and cleaves it into single amino acid, and you get rid of those proteins you don't want. But it turns out that pathway was not upregulated in our mutants, but rather the pathway that makes the immunoproteasome. So the immunoproteasome is a specialized proteasome that was first found in antigen-presenting cells. So those things are going to be part of your immune system. They have 5 proteins that are unique to the immunoproteasome. They do not rely on ubiquitin. They do not rely on ATP. And they cleave in disordered regions to give you pieces of protein that actually can then be presented in an immune cell as an antigen. And that turns out to be the pathway that's upregulated. And it's also mislocalized. So instead of being in the cytoplasm here in the magenta, it now is residing right under the cilia, where all the proteins that didn't dock have now gone. And that's probably where we think they're actually being captured and partially destroyed. And so we're now treating with drugs that are specific for the immunoproteasome, and it actually, in cultures, gives us an improvement in some of our phenotypes. So we're really excited that we might have a way to actually help some of these patients. There turns out to be a second motility-independent phenotype, and that is the amount of mucus. So this is a section through normal lung that we get by transplants. This is a section through CCDC39 cells, and the magenta is mucus. This is MUC-A5C. So you can see a little bit here in the controls, but there's massive amounts. And they make these giant mucus plugs. And so the cultures that we're looking at have ciliated cells, they have secretory cells that are making mucus, and when we look in cultures, we find that the cell fate appears to be changing. That normal cultures have very little purple. It's probably hard to see magenta. These patients have increased amounts of mucus-producing cells, shown here in the graphs, but other PCD patients don't have this. And the decision between being a secretory cell and a ciliated cell is regulated by Notch, and Notch turns out to be inhibited by a small molecule called DAPT. And so if we take and treat our normals with DAPT, there's really little difference. But if we treat our patients' cells with 39 and 40, we go from lots of mucus to removing it. Now, we were really excited because it meant, and we've now shown, we think that the cells actually are multiciliated, are transdifferentiating back to being a secretory cells, and that we can again change the pathway by affecting Notch. And so, I just want to end by saying we think that evolution, because it has such a wide number of organisms, it gives us a lot of information about key genes. We think we can use model organisms in some ways to have really fast, easy therapeutic assays, and that the human variation provides us with the ability to think about phenotypes and potentially new phenotypes and mechanisms of disease generation. I went a little long.
Well, thank you for that magic talk. We have one question there.
Really liked your outline. Have you looked after the 9/11 events, whether patients, fire department, exposed to the
That's
mutations and have an altered mortality rate as a result of that? Because they are sick-cell.
It's an interesting idea, but we don't know. There's lots of studies about particles and then their effects on lung function. We have a project looking at viruses in lung function, but we're not very far into those yet.
Second question.
Question there. Now use the microphone, please. It's just behind you.
Can I do one more?
Not without the microphone, you can't. There's. Oh, go for it then.
Microphone is functional now.
Thank you.
That's a joke. Can you explain what happens with hydrocephalus? That was between parentheses in the different, in one of your slides. What is the link between the PCD mutations and hydrocephalus?
So.
You have to explain that question.
Okay.
Yeah.
So. Cilia genes have been suggested to be involved in hydrocephalus, and that comes mostly from model organisms. which turn out not to be a good model for humans. And so it's thought that in mice and in zebrafish and in xenopus, you make mutations in any of these cilia genes, you get hydrocephalus. And it's thought that it's because the fluid in the ventricles is not moving properly. In humans, that doesn't happen. And it's thought that the reason is that the diameter of the spinal column relative to the brain means that you don't have this pileup of fluid and the increased level of increased fluid in the third and fourth ventricles.
So with congenital hydrocephalus then, where sometimes the canal of Sylvius is not functioning properly, there are no correlations to mutations?
There are
actually,
very
is that
few.
correct? Very few.
Hydrocephalus, the major hydrocephalus that are actually treated in clinics often come from brain bleeds. And iron turns out to have a major effect on cilia. And so many of those may be indirectly related to late onset of it, but early onset doesn't affect hydrocephalus.
Thank you. I just have one follow-up therapeutic question, and that is, Vertex make quite a lot of money out of their CF drugs, and DNase is another drug that they use. When you're thinking about mucin and cilia, what would you do if you were Vertex to try to affect that axis?
So the Notch inhibitors would be an amazing way to think about doing it. The problem is that Notch inhibitors have major effects in other organs. They wipe out your gut.
They're not good.
And so there's a group in Germany that's actually trying to develop lung-specific Notch inhibitors, and so we're really excited about that.
Very good. One there. Microphone, please. It's coming, it's coming. You can't shout loud enough.
Tom had his hand up first, I think.
Well, he's won the house one already, so he'll have to wait.
No, I haven't. I didn't get to ask one last time, and I haven't asked one this time.
Okay, go for it then. Sorry. No, let him go.
So
first.
after
It's
Rich's
okay.
talk, it made me think, about suppressor mutations, because that's what he was talking about. Can you select suppressor mutations in CLAMI, and have you done it? Because I would think you could come up with all of these pairs that way.
We have amazing numbers of suppressor mutations. I would be happy to tell you. So we've done it a lot with the CLAMI mutant in CCDC39. We have the one that's the best is we have one that's actually in the C-terminal tail. It actually makes the protein temperature sensitive. So it's completely functional at 21 degrees. And when we shift it to 32, they look like the null allele. And so we've isolated suppressors of that, and we get mutations that fall into CCDC40 and as well as 6 other genes.
And can you go from that to drugs you might use in human patients?
The problem is that they're specific for the TS allele. So when we do the suppressor analysis in the null allele, which are almost all nonsense mutations, we either get the nonsense going back to another amino acid. And so I would love to be able to do that, and we've tried hard, but it didn't work.
Go for it.
I really enjoyed that talk, Susan. I wanted to ask, what sets the length of the cilia, and is it known? And does the length matter? Because it seems very preserved for...
Length turns out to be really important. Yeah. Short cilia actually have defects, and long cilia have defects. So it's important to actually monitor length. And especially for sensory cilia, length actually ends up having lots of phenotypes. And there's a complex of 4 proteins that include MAP kinases that seem to be important for making decisions about length. GSK has also been implicated in length control, and so there are lots of different ways to affect length.
Okay, thanks for that. There's one in the corner there which I missed because the light was in my eye. Go for it.
Sorry, this is...
No worries. Since there's new research showing evidence for possible secondary ciliary dyskinesia and cystic fibrosis, would this research help with mucus clearance for CF patients?
So CF is a really different disease. CF is really fairly well treated by this 3-drug cocktail now that Vertex makes. But CF is a disease in which the mucus is really stiff. The cilia, the mucus is here, the cilia are here, and so the stiff the cilia don't really impact that, and so it really is a very different disease.
Okay, thank you. Go for it.
Sorry, I'm a very much a lay person. I just have two questions.
I
One,
apologize if I was above.
oh no, you were brilliant. And this is my 17th time here, so nobody's. So I have, no, maybe it was 16th. I think I missed one. But just two quick questions from a lay perspective. I've worked with severe chronic anemia patients. And the question is, patients who are iron depleted, is this something that also people should be considering in relation, your work, in relation to anemia, severe chronic anemia, and iron depletion? That's the first question. It may be totally irrelevant.
I do not know the answer to
Okay.
that.
The second question is a very different one, which has to do with our environment and what we're bringing into our bodies, and microplastics. Is that, from your perspective, an issue?
Yes, so I mean, there's lots of these particles, both macroplastics as well as things from the 9/11 things, just basically particles actually end up affecting these cells a lot. And there are people that are starting to look at those and trying to, but it's really hard to get any feeling of what to do about them.
Sure.
Great. Well, we've got one more there, and we'll finish up there. Do you need the microphone coming? Yeah. So two more, that one and then this one. Hold on, let's do the one at the back first, and then you.
Thank you, Susan. Two questions, if I may. You mentioned in passing the correlative microscopy and the dots. Can you say a little more about them?
The what?
You mentioned the correlative microscopy and the proteins you identified that way. Can you say just a little about those? And secondly, I'm curious, you described more or less a linear addressing scheme. In an assembly that complex, do you also see instances where the addressing scheme recruits one protein and then that recruits another, and that, I mean, I would be surprised if it was strictly linear.
So for the dining arms, because they're actually made in the cytoplasm in those structures I told you, and the radial spokes are also made in the cytoplasm, they get transplanted as whole complexes. But there was another structure called the NDRC. It actually moves up individually. And we don't know very much about how that happens. And so they each move up and actually somehow are assembled within the cilium, which is, I think, another big issue. I mean, go ahead, you had another question.
No, the other question was just, you mentioned the light microscopy. Can you say a few more words about that? Because the images were amazing.
So we have switched to using both expansion microscopy, where we treat them, it's sort of, I like to think about it as a baby diaper, the way if it gets wet, it expands. You can sort of do the same thing for cells, and we can get 4x or 8x, so we can get above the light resolution image by using expansion. But we also do STED microscopy, which is a super resolution way. And we now, one of my pictures had little green dots I showed you along there. We're seeing those things at 96 nanometers with light microscopy now. And so we can now go in and actually look at the light level for proteins that we know about and actually ask where they are. And I think it'll give us a much bigger handle for trying to understand how other proteins are addressed.
Great. I think we got time for one more. Was there one more at the back?
Yeah,
Go for it.
Hi, I'm wondering if you've done any cryotomography and if the structure of the cilia is the same from the base to the tip or if it varies.
So we haven't done cryotomography, but Danny Nicastro, who was actually a postdoc with Dick McIntosh here to begin with and is now at Southwestern University in Texas, has done a lot of tomography. And that's the other part of the whole thing that I didn't tell you, is that I told you about all the things that are the same. It turns out the base is different from the tip. It turns out that the 9 doublets each have some of things that are unique about them. And so tomography is the way to find those things. The single particle cryo-EM, because we lose spatial resolution in that respect, we just average everything. The tomography keeps everything intact, and so you can actually ask about what's happening at the base, what's happening on doublet 7 versus doublet 4. And so tomography is a perfect correlate to doing what we do.
Well, thank you for that. Thanks for your talk. Great, really enjoyed it.